Good morning, students. This is Dr. K Surendirakumar, Assistant Professor, Department of Microbiology, JJ College of Arts and Science, Pudukkottai. Today we are going to talk about radioimmunoassay. It is one of the antigen antibody interaction test. The radioimmunoassay is one of the sensitive immunoassay techniques. It will help to determine antigen antibodies in a sample using radioisotopes. It is a kind of in vitro test. It will be more specific to quantitative detections of antigens. The RIA was first described in the year of 1960 by Burson and Gallo. Radioimmunoassay technique is help to determine the concentration of the particular antigen in a sample based on the competitive binding between unlabeled and labeled radioisotope antigens in a specific antibody. It is important tool in biomedical research as well as clinical practices. It is commonly used to determine the microbial antigens, proteins, vitamins, hormones or drugs in a serum in very minimal concentrations. RIA have three major principles. One is immune reactions like antigen and antibody binding interactions. Second one is a competitive binding. Third one is measurement of radio emission. It have certain requirements. One is radio labeled antigens is called as hot antigens. Generally the gamma rays emitting isotope such as I-125, beta rays emitting isotope such as tritium. Second one is specific antibodies. Third one is unlabeled antigen like sample antigens is called as cold antigens. Fourth one is microtiteroplate. It is 96 well plate, commonly we call it as ELISA plate. And fifth one is washing buffer. It is 1% trifluoroacetic acid. And what is the procedures? First, we need to prefer radioactive antigens along with antibody. This is first primary antigen antibody complex. Then later we need to add unlabeled antigens like sample antigens, because here we are going to test whether the sample antigen having specific antibody or not. Okay. So this after adding the unlabeled antigens, what will happen? Because primarily we have one antigen antibody complex, that is radioactive antigen as well as primary antibody. After adding the unlabeled antigens, the radioactive antigen is displaced by unlabeled antigens. So what will happen? The precipitate of antigen antibody complex with anti-immunoglobulin like a secondary antibody. Then radioactivity of the superintendent. So which radioactive antigens is displaced by the unlabeled antigens, that will be washed out. So the radioactively of precipitated of it means the bound antigens. The bound antigens means what? The unlabeled antigens. Now it will be labeled by radioactive antigens. Is called as secondary antibody. So this RIA having two different methods. One is double antibody RIA. Second one is coated tube RIA. In the case of double antibody RIA, we need to add first antibody as well as radioactive antigens. Then we have to add unlabeled antigens. Then unlabeled antigen, what will happen? It will be displaced by radioactive antigens. Then the radioactivity of the superintendent solutions will be removed, and that radioactivity of the precipitate, it means it will form a secondary antibody. Secondary antibody. Then one is coated tube RIA. So in the coated tube RIA, the primary antibody is coated on inside the each tube. Okay, first we have to coat it with the antibody. Then the unbound labeled drug, it means that sample 1. Sample 1 can be easily removed after pouring it. When that primary antigen antibody will not form a secondary one, it will be removed by through the pouring of the supernatant. It is called coated tube RIA. It has certain applications. This RIA is used to detect the peptide hormones, different viral antigens, hormones, drugs, hepatitis B surface antigens, even mycotoxins, and early stages of cancer we can detect it using RIA. And what is the advantages? It is high specificity, even minimal concentrations, even 0.01 mu L or mu G concentration we can detect it. Then high sensitivity. High sensitivity, it means if we have the antigen, means if the specific antibody is there, then only it will be bind, otherwise it will not be. The third one is even a very small amount is enough to digest. Then it has certain limitations, because in this technique we are using radioisotopes, so we need to be risky. Then after tested it, the disposal of radioactive substances is problematic, and because we are using radioisotope, this test will be costly and expensive. And fourth one is the radioactive substances have short life. So these are the certain limitation is there in the RIA case. So RIA is a type of antigen antibody interaction test. Second one is it is in vitro test. Even in a minimal concentration of antigen or antibody, we can detect it. We are using radioactive isotope. Here primary antibody and antigen, unlabeled antigen like sample, then it will form a secondary antibody complex. Then the fluorescence emission is there that we can analyze. Okay, thank you.